Very good morning, ladies and gentlemen. Professor Ganguly, Larry, Walker, Ram, Vishwakarma, Professor Handa, many other distinguished invitees, speakers. This is a great pleasure for me, as was reminded by Professor Ganguly, that two previous directors and one present director is here. Before I started, I would like to recall about 16 or 17 years back when I was working as an enzymologist, biotechnologist, as he mentioned about me. I had never ever dreamt to be the director of, at that time, RRL Jammu. I was traveling from Hyderabad to Delhi, and Ganguly got up and came to my seat, near my seat. It's about 16, 17 years back. And to tell me that I should not aspire to become the director of Institute of Microbial Technology. I was biochemist, microbiologist, fermentation man. That's what the IMTech was actually built for at that point of time. But he said, I don't want you to become the director of that place. Well, I said, that's the only logical place where I should go. He said, no, Jammu is the logical place for you to continue there. I'm saying this only after 16 years. I'm reminding him that he's also some kind of, I must say, astrologer, perhaps. And it was only after 2 years that I was given this assignment to be the director of that institute. Let me say that I knew nothing about plants as drugs, or I knew nothing practically about pharmacy, because I was a biochemist. And so the colleagues in Jammu showed me the way when I joined, because this responsibility was given to me, and I forgot about my science, whatever I had done. And today, after 5 years of leaving Jammu, what I'm telling you is bringing you back to those the times when I was at Jammu and how I built, what we built, and this all work, what I'm going to say is show as examples is done by people at Jammu, and now in the last 5 years at Jamia Hamdard, where I moved. So I think I have been also assigned for the last few years, thanks to some people who thought that I could take that responsibility as a member of as a chairman of the Pharmacopoeia Committee, Unani Pharmacopoeia Committee. And so therefore I had to learn more about pharmacopoeias, and as well as bulk products that make part of these pharmacopoeias. I'm also a member of the Ayurvedic, I would say Pharmacopoeia Commission that the country has now established, and Professor Handa is looking after that. Professor Handa was also looking after Ayurvedic Pharmacopoeia concurrently when I was looking for a Unani Pharmacopoeia. We worked very closely on those issues. So when I was repeatedly, reminded by my colleague, Dr. Bedi, that I should come here, and I thought it was an opportunity. After 5 years, they remembered that I should be also part of some discussions with IIIM. So I was wondering, what should I talk about? Then it came to my mind one day that I could talk about the scientific pursuits that actually happen in most of the institutes within CSIR and outside CSIR, which actually we leverage at some point of time for pharmacopoeia. Even the pharmacopoeias, and which we built over the years, many people think that they should not be dynamic at all. You just develop it and then leave it there for companies to follow. But we know in the case of plant, science, and in the case of traditional medicine, we will always keep on developing something new depending upon, the need of the time. So therefore, I thought about this. As you all know, because most of you come from the background of Indian systems of medicine, I'll say there are some challenges which we know when we build a pharmacopoeial monograph. We know exactly what are the challenges. So you can see, I don't have to go through each one of these lines which I have put there basically because I have almost 50 slides, so I will definitely have to walk through these. And I think some of the problems which stare on our face is the documentation actually. Even though we say we have 5,000 years history as far as Ayurveda is concerned, and we have hundreds of years of history as far as Unani medicine is concerned, but I must say that for the regulatory point of view, we still have, we still do not have proper documentation. And the last, line, which is very important, is that we are expected by our regulators, when we talk to them, I happen to be part of Indian Pharmacopoeia also in the advisory council, and always we are talking about comparing these with the single molecules. I think I was very glad that Ram touched upon this, that even though as he was defined as a hardcore chemist, I would say that he talks about the multiplicity of the molecules that we have here. Just a glance of what is the industry today as far as the traditional medicines are concerned. We have almost about 8,000 manufacturers in this country. I'm sure some of them are sitting here, but you can see there are only 10 companies which are over $20 million US. And the bulk of the companies are very small, maybe 1 crore equivalent or 1.5 crore equivalent. But you must also see the strength that we have is we have 500,000 medical professionals here. This is the volume of the Indian system of medicine, almost 1 billion, which is recorded here, but we have export of extracts and other things to the extent of about 600 million. Yesterday, somebody was telling us about isabgol touching already about 100 million or 150 million US. So what is the regulatory control on these in the Indian system of medicine? I just enumerated the historical perspective, what is regulating our Indian system of medicine. And you will see that a landmark decision was in 1970, where we realized that we should have a separate, although Professor Ganguly said that this is perhaps becoming a big issue, unless and until we work all together, it's going to create problems. But we are actually controlled under a different system, not as a part of the conventional medicine. So you can see there are how many companies are actually registered already in this country. And you can see that we have established referral traditional hospitals in this country. So this regimen is up and kicking, and it has to build up, and it has to be most modern. So Pharmacopoeia Commission, which came into being, and Professor Handa is actually spearheading most of the work of Pharmacopoeia Commission. I think that is something which is now going to be a single window where most of the issues with respect to the static or dynamic nature of the traditional medicines will come up and will be sorted out. And we have Ayurvedic pharmacopoeia. There are several committees which are looking after this. You can see the monographs, keeping on, adding on to these monographs which are already available to us. What is the quality of these monographs? What is the quality of the requirements that is there is a debatable issue, which of course I will touch upon, and which is where the dynamism has to come up. So we have also a Unani Pharmacopoeia committee which is looking after the Unani side of this, traditional medicine paradigm. So generally, as most of the people know, that the focus of the monographs that we have is the physicochemical parameters, limited, what is called as proof of the product, and it is basically because what I showed in the first or second slide, that we are practically keeping in mind the 7,000 companies which are actually the backbone of this particular drug industry. So, but at the same time, I think the traditional medicine education, as I showed you, that there are 500,000 practitioners already registered, and there could be many, many more who are not registered, and we have a proper, strong education. My institute is running a Unani, education, and it is also running modern medicine education. So both of these are running parallel, and there is an overlap being created between the two. That was what I had dreamt of when in Institute of Integrative Medicine was created out of Regional Research Laboratory. What Professor Ganguly mentioned was the TKDL, which actually helped putting all the resources, earlier resources that we had with us, at one place. So what is the actually the quest is to make these products better, safer, and we must develop them faster, and most importantly, affordable. By the way, today, any herbal drug, or for that matter, even the combination drugs or Ayurvedic or Unani drugs, are not affordable at all. It's a myth that they are affordable, but this is what would be one of the important functions that people have to. Some recent initiatives that we were part of. One was the TKDL, of course, and the other was the Golden Triangle program of the CSIR and the Government of India, where we try to bring these people together, traditional doctors, modern practitioners, and contemporary scientists, and to understand, first of all, where the link is missing, and then the extracts to replace the churnas. That was actually a very big issue because the bulk of the drug was becoming a problem. And then reverse pharmacology approach. I think this came up for first time from India, where we said we do not have to follow the modern drug approach as far as the creation of clinical evidence is concerned. So I think these are very important issues, but I think one most important issue is the registering of your clinical trials in the country. Actually, people would go for clinical trial or clinical evidence development without a person next door knowing what is happening. But now it's not possible, so we have to register all the clinical trials accordingly in an ICMR registry. That's what is, I must consider this as one step forward to regulate it appropriately. Then I think I'm coming to the actual subject now, is the standardization and the quality control. And I must say that a lot of efforts have been put in the country by various companies in private sector and public institutions A lot of work has been done and which is accumulated over the years, but we have not been able to put them into a proper focus as far as eventually drawing things out of this effectively to make these as the parts of the monographs and parts of the pharmacopoeia. That's what I thought I should convey here when I come to give this lecture. We have to also take into account the traditional documented plant parts which are generally regarded as safe. I think that makes our life a little bit easier to move forward. But it's an issue which is highly debatable because we do not have any pharmacovigilance as far as the traditional medicine is concerned so far in place. And whenever we talk about pharmacovigilance, this word vigilance becomes a little bit of an issue. And I was always telling the authorities that can we remove this vigilance word and then perhaps the acceptability will be much better. So then you can say that authenticated by the identity of active substances is something which everybody is talking about, even though this has not become practically a requirement today as far as the pharmacopoeias are concerned. So raw material control is one issue which we have to take in account. And not only raw material control, but the product that we talk about taking to the clinics or for creating clinical evidence. I'm very glad Ram mentioned this morning that the plant, which we have, the production plant, which I had initiated about 6 years back in Jammu to make a good manufacturing practices for small quantities, up to 5 kilograms maybe, for those products which will go for clinical trials. But the biggest issue is a lot of clinical trials are being done individually by people, but we cannot trace back and say where the material came from, what was the quality of that product which has gone into the clinical trial. Even though we make a dossier, we do mention these things into this, but in real sense, it's becoming difficult. Even I know in my times when I was working there, if we wanted to get into the clinical trials of the things, we had to approach some of these big companies, say extract companies, Amit is sitting here, many others. It was always becoming difficult actually. We cannot expect a company to stop production, clean the whole thing and then produce a 5 kg for us, 5 kg material for us. So at that point of time, we thought that we should have it right in the institute itself. I'm very glad that it's coming up, as he mentioned, that maybe 6 months to 1 year, it will be up and running, and many people should actually try to use that eventually. So these are good agricultural practices. I think we have been talking about it for 20 years or 25 years in this country, even though we have a medicinal plant board, they have their regulations and all that, but we should not, forget that more than 80% of the plant material that is being used in the herbal products is not actually manufactured or through agricultural practices. It is extracted from the forest. So I think if we can go into the good post-harvest technology, harvesting times, processing of the plant part. I, by the way, learned all these from Dr. Handa, because when he was the director there, he was actually looking at these aspects very closely. This is a referral standard. This was built by Dr. Handa in Jammu, and where we had hundreds and thousands of materials kept there as the referral materials, and this was in fact the base when we started thinking collaboration with Mississippi, that we said that this is a base available with us which we can perhaps share. And so I think this has now come up in various places in this country, which is going to be a watershed as far as the future quality of these plant materials are concerned. What is still being not practically adhere to is the manufacturing controls. That comes through the good manufacturing practices. We have in place GMP regulations as per the government. Every company is supposed to be following good manufacturing practices. But whether we are following these, that's why I said the documentation is something which is still not connected completely to the ground situation that we have. We are very concerned about the heavy metal analysis, microbial loads, and so on and so forth. So this is the standard platform which generally every plant, chemist or or a traditional medicine person who wants to get into the modern mode would like to follow. And I think this is what I had established in Jammu when I was the director there. And believe me, most of the things will be thrown up from this platform. We can have the extracts, fractions, molecules, then in vitro, in vivo, screens. It was talked about the, the online testing and high throughput or low throughput or medium throughput testing. This is what at that point of time it was in the back of mind when we developed these protocols at that point of time. This can give you a product, a new herbal formulation. It could give a new lead molecule, and you can optimize it. It can go to a botanical additive. It can go for a clinical trial, and so on and so forth. I think this is practically what everybody is following. Just a collage to show you what was created at that point of time, because Professor Ganguly mentioned that we still do not have that quality of research backup in the country as far as the botanical drugs are concerned. But believe me, what I am showing is about 10 years back when, maybe 7, 8 years back when I was still at Jammu, when we had created almost everything which was responsible to enhance the quality of, welcome Dr. Trehan. And what I'm showing is what has been created in Hamdard University after I joined here last 5 years. And you can see this is by and large not only replicating what we had in Jammu, but we are actually taking it to the next height. And you can see in analytical tools and also the possibility of carrying on these researches which will actually contribute to the dynamism of the pharmacopoeia. I think I invite you all to come to the institute and have a look at these things, and most of these instruments are actually used for herbal products or I would say isolated molecules, natural molecules, so on and so forth. I think after having shown this, I would say I will have to skip some of this. But what is most important, slide which I was always excited about when I was in Jammu was when we are talking about the assurance of the quality and consistency of these products. That is where we have practically failed most of the time. I remember in CNN I saw once a piece, and I was excited that they were showing a product, a multi-component product of India, which has withstood the times. And I don't want to name the product. And then ultimately the last line in that, and also you must know what is the rest of it, because the impurity profile will be there. In a botanical drug, it is the reverse actually. You may be only needing 0.01%, which you should know exactly what it is. And 99.98 perhaps or 99 is not what is my concern. So, but if you can create a profile like this from batch to batch, which may not be actually superimposable correctly, but this is what would be my dream to have in a pharmacopoeia. That is what is called the dynamic pharmacopoeia, not for 4,000 or 7,000 companies which they have to put their products in the market. Well, they should follow the pharmacopoeia that is existing today, which is which is an approved pharmacopoeia. Lot consistency can come only if we have to actually work for the manufacturing of these, taking back to the source of the plant material that we'll have to take. And what is one intriguing issue which everybody is thinking today about is stability and shelf life of these products. I think we're discussing it off and on in our pharmacopoeial committees or pharmacopoeia commission, and unless and until we create actually a component of quality control and quality assurance to the strong level that we can, clearly say what should be the stability, what should be the time for which this particular drug has to be used. Sometimes we follow classical preparations. There are some classical preparations where it is perhaps not required to look at the shelf life if we are talking of metal compounds or basmas or pushtas in the United system of medicine. I'm sure the life will be perhaps 10 years, 15 years, 20 years, you don't have to bother about it. But we must define actually how to keep them, how to put them into place. I think, Bedi, please do tell me that if I'm taking too long. I think ashwagandha was something which Ganguly mentioned. I thought he will stay on and I can remind him of the quantity, the work that was done by Jammu, I think. I think it has been acknowledged, it is published, it has been used by many people. We have identified, I must owe this slide to Bedi because in his department this was all being told to us that different varieties, what kind of, how you can make a distinction about it. Even though today also ashwagandha is ashwagandha. People say, well, it is ashwagandha, but I can show you in the couple of next slides that the varieties that we got, cultivars that we got from throughout the country were absolutely not superimposable. And so we did a lot of work to look into, to identify, this is simple HPTLC profile, which of course is also propagated through in our pharmacopoeial standards. But you can see the difference between the various varieties. And what Professor Ganguly was mentioning about actually going deep into and identifying each peak in an HPLC. And I must owe this to a wonderful natural product chemist that institute had in Jammu. And perhaps we did not see any parallel of these anywhere in India. I'm proud of saying that. They were not built by me. They were built by visionaries much before I came there. But they did such an excellent job that even US Pharmacopoeia had to acknowledge and take some of these reference compounds from us, which have become part of US Pharmacopoeia as far as Withania somnifera extracts are concerned. So you can see the beautiful separation that was worked. It doesn't happen overnight. It took years actually, to tell you the truth. But everything was rewarded, everything came in good publications. If you see MS of these, you can see Withania's molecular mass of 5 compounds the same, and but MS could tell you exactly what is different and how we can actually identify these. I'm sorry, I'll have to move on basically because I don't want to take more time. So what is also a debatable issue, I'm sure Professor Handa will not accept it at this point of time basically because it is not a part of our pharmacopoeia. That can we see the potency through biological activity? I say future, perhaps 10 years ahead of now, we may have to follow these as the dynamic part of the pharmacopoeia. Bioavailability, degradation products, I think these are very essential which have to be established through biological activities. One slide which actually I always discuss about is, although it looks very simplistic, representation, but if you can see, if we can say if there are 3 molecules as the marker compounds in Withania somnifera, and we say this is the range that we should have for vitaferrin A, because vitaferrin A is very toxic otherwise. I have varieties where you have very high content of vitaferrin A and very low content of vithanolide A. You can see the difference here. It's 5 to 10 times more, but that is what is the requirement. And this is not based on simply chemical analysis, but this is based on biological activities which have been worked on in vitro as well as in vivo in Jammu. I think these are things that I wanted to talk about. In summary, I must say, botanical drugs should be standardized based on identity, potency, purity, and consistency as well as stability. This is very important. Then equivalence between the products of different manufacturers claiming similar botanical components required to be established. You cannot just say there is, this plant material is in this, so therefore it is the same product from one company to another. And contemporary approaches for quality assurance of botanicals are essential for long-term benefits of the society. I think in essence, I have said what I had to say today, but I will perhaps not stop before saying, giving some, I say, precedence to my own institute now here, where we have we have a very strong groups. I think a couple of them I just noticed sitting here, where we have analytical methods developed for which, in my opinion, should become tomorrow's part of the monographs that we'll be producing for our plants for a better, safer, and efficacious products. So you can see these publications which have come up. I can leave this with Dr. Bedi, who all those who are interested in looking at these can look at it. Some of the wonderful publications that have come in the last 4 or 5 years, and which are actually happening very fast at our institute. And there are also biological screens set up for various activities in vitro, in vivo, and also every component which is got obtained as a single molecule or as the complex molecules, druggability is being established, and you can see the examples here where of course they are going one step ahead. It's not a part of any pharmacopoeia, but they are actually talking about the nanoparticles, solid lipid nanoparticles, which would have shown extremely different kind of profile as far as the activities are concerned. And so there are other studies that are in progress. Perhaps, I don't know whether some of my colleagues are going to make a presentation here, but all those who are interested in getting little more information about this may visit our website or may actually come and walk into our labs. I think marker compounds was mentioned by Dr. Ganguly. This is a very, very essential. I can see some of the extract companies, Doyne sitting here. I think whole country has to actually focus on these. When I was in Jammu, I think I had built up about 120 such molecules and put into the website, and we were of course also selling it abroad to US and to other companies, but I'm not sure what is happening now. So I think I just wanted to stop my presentation with this reference, a publication which has come in January 2013, which is actually, if you look at it, this was part of the research and Nimitli program of CSIR, and they came up and they said there was a huge clinical research done where they wanted to actually see the alternative to glucosamine and celecoxib. And this presentation, this publication from Arvind Chopra came up very well, and it is highly rated paper which has appeared only less than a year back. And they, of course, acknowledged the contribution of CSIR and my lab where I was working at that point of time. So I'm very glad that I got this opportunity to come here and to present whatever I thought is very essential as a starting point for all of you to deliberate upon for next 2 days, and say that unless and until we think about the standards and pharmacopoeial standards of these drugs, which we may have faith about, but we have to actually talk about those who do not have faith about them, and we have to prove it to them. Before I end up here, before I say that I'm thankful to IIIM that they gave this opportunity to me, but I'm also thankful to Mississippi Group because the journey with them started almost about 11 years back. And I highly appreciate the perseverance of, because everything does not always happen the way we want. And there are several things which we could not keep the consistency upon. But that's the way we work in India, and that's the way. And so by now you must have realized that you have to be actually prepared for those eventualities as a part of the system and then move forward. I'm so glad that they're still ready to come and be part of us, even though for last 2 years there was almost, I thought that this whole CRISM concept was already given up. But since personally, I don't look back. When I leave something, this is the new leadership which has to take it forward. I don't even comment on that. But I am very glad that when I found that there is this kind of a forum created, and I'm welcome in that. That's why I'm just saying I again welcome you back to our fold. And Larry, I think I have great regard for you for being patient with us and also being consistent. And lots of people from India have gone there and got trained who are part of these all developments which are taking place. Thank you very much for that contribution to India. Thanks a lot for giving this opportunity to me.